The California Natural Diversity Database, also known as the CNDDB, is the Natural Heritage Program for California. We're a program within the California Department of Fish and Wildlife and have been around for 40 years now. There are similar programs across the world which are coordinated through an organization called NatureServe. So what are the problems that natural heritage programs were created to solve? What is the CNDDB and why does it exist? A group of conservationists had a mission to prevent the loss of biodiversity. They ran into a problem, though. There was a lot of biological data around, but no one-stop shop for all the information. You can't know where to focus your conservation efforts if you don't know which life forms are imperiled and where they're located. The solution was the creation of regional heritage programs to gather, analyze, and distribute information regarding imperiled elements of biodiversity. They began by partnering with private and public programs in each state in the U.S. Those efforts were later expanded to include Canada and Latin America. Natural heritage programs now assemble and distribute information on biodiversity across most of the Western Hemisphere. Imagine you received a large quantity of mixed data. How would you make sense of it all? Here we have gray circles representing an array of biological data scattered across a landscape. To prioritize conservation efforts, we first need to identify the most rare and vulnerable things. We then categorize everything into elements of biodiversity. These include individual species as well as natural communities. Next, we need to distribute this information to people who can use it for conservation and management. The CNDDB has two stages of data processing and sharing. The first step is rapid but imprecise. We review all our incoming data and determine which counties and USGS quads a species is known from. This information is distributed through our Quick View tool. Due to time constraints, some species we track are only mapped at this level of precision. Our second and more precise step of processing is to map element occurrences. Element occurrences are map features which summarize everything we know about an element in an area. They're created by assembling data and mapping that information while incorporating the uncertainty of the location. The majority of elements we track are represented by element occurrences. In review, an element of biodiversity is a plant, animal, or natural community tracked by a heritage program. Most elements are plant or animal species, but since heritage programs also track natural communities, we refer to all the entities we track as elements. An element occurrence, also called an EO, or an occurrence, is a specific location where an element's been known to occur. We don't map repeat visits to a site as overlapping features. Such information becomes summarized into an element occurrence. We modify EOs as new data is received for a site, so EOs can change in size and shape over time. So what types of elements are tracked by CNDDB? Currently we track about 2,400 types of plants, about 900 types of animals, and for the first 20 years of the CNDDB's existence, there was an effort to map occurrences of rare natural communities. That work's been replaced by VegCamp, which maps all vegetation types, not just rare vegetation communities. VegCamp is a separate program that maintains their data outside the CNDDB. Users today will see that 3% of the occurrence records in the CNDDB are for natural communities. Please be aware that these community occurrences haven't been updated in 20 years, and they don't follow the same standards now in use by VegCamp. The CDFW Vegetation Classification and Mapping Program, or VegCamp, follows the national standards and maintains the vegetation classification system for California. They use ground surveys and aerial photography to create detailed vegetation maps. Communities are given ranks for rarity and threats, similar to the way species are evaluated. The mapping of California's vegetation is an ongoing process. This effort is led by CDFW, along with the assistance of multiple partners. Areas shown here in green have already been mapped by VegCamp. You can find these vegetation maps in BIOS, which is our department's web map viewer. For many areas outside of where VegCamp has worked, you may find that the old CNDDB community occurrences are the best data available, but be aware that these will be superseded at some point in the future. We don't have time to go into detail about VegCamp during this talk. If you want more information, just do a web search for California Vegetation Mapping or VegCamp, and you'll find a wealth of information on their website. Switching gears back to CNDDB's current work. There are two main ways we determine which elements we track. Remember, we want to focus our efforts on collecting and mapping data for the most imperiled and rare species. The first way is by referring to official lists of imperiled taxa. CNDDB tracks nearly all species listed as endangered, threatened, or candidate under the state and federal Endangered Species Acts. We also track animals on the California Species of Special Concern lists, as well as plants with the California Rare Plant rank. Our second method of determining what to track is through evaluation of imperilment. Experts may come to us with findings that indicate certain species are in decline and warrant our attention. Natural heritage programs use a standardized ranking calculator created by NatureServe to evaluate the rarity of an element. Elements that have a state rank of 1 to 3 are tracked by the CNDDB. The CNDDB website has links to the Special Plants and Special Animals list, which provide taxonomic and ranking information on the elements we track. So how does this element ranking number system work? The numbers range from 1 to 5, with 1 indicating the greatest level of imperilment and 5 indicating the least. The global rank, or G rank, describes the condition of a full species in its worldwide range. The taxon rank, or T rank, is used when an element we track is a subspecies, variety, ESU, or falls into some other category below the full species level. The T rank describes the condition of an element in its worldwide range. Finally, the state rank, or S rank, indicates the condition of an element within California, whether that element is a full species or is subspecific. As an example, let's look at Coryphantha vivipara variety rosea, an element ranked G5 T3 S1. This cactus is a G5 because the full species, Coryphantha vivipara, is a common cactus found from southern Canada through northern Mexico in the area in orange. It's a T3 because the variety rosea is restricted to a few mountainous sites within the deserts of southeastern California, southern Nevada, and northwestern Arizona, inside the pink area here. It has an S1 rank because in California it's only known from a few occurrences near the Nevada border in the small area shown by the star. So where do we get our data? CNDDB staff seldom conduct our own fieldwork. Instead, we rely heavily on the work of biological consultants, agency biologists, academics, and conservation groups. We gather specimen data from museum and herbarium websites. We even get data from citizen scientists through CalFlora and iNaturalist. CalFlora has a form in their app that contains fields the CNDDB is interested in. We also have an iNaturalist project that people may use to submit their data. If you or your colleagues run surveys for rare species, please report your findings to us. We won't be able to share your data if we never receive it. What types of data does CNDDB use? We receive about 1,000 data sources each month. We currently have 250,000 of them in our files. Since we accept data in many formats, there are wide variations in the amount of information we can glean from sources. For example, a CNDDB field survey form typically focuses on a single detection of a single species. These simple sources can be reviewed and mapped quickly. On the other hand, a written report or database may cover many detections of many species across a region, and such complicated sources require substantial effort to add to the CNDDB. We also use sources such as emails, published articles, and books, which all require staff time to sift through and interpret. While there are many ways people submit data to us, we prefer to receive data through the CNDDB online field survey form. This form is actually a website. Users log in, choose a species from the list of everything we track, and are asked a series of questions relevant to that species. Locations are recorded through a map viewer. Other supporting files, such as photographs, spreadsheets, PDFs, or GIS data, may be uploaded along with each record. Users can get reports on their previous submissions to prove that they sent their data to us. Online field survey form records are saved directly into a spatial database, so they can be shared with others without CNDDB staff first needing to interpret them. Raw data from online submissions are made available to CNDDB users within a month through the BIOS viewer. To access the online field survey form, or to get guidance on submitting other types of data, please visit the CNDDB website. How do we keep track of all the data sources in the CNDDB? Every source with useful information is given a unique identifier, called the source code. Here we see an element occurrence record. Zooming in, we can see a list of citations for the EO. This includes a source code and a brief description for each source. If you have questions about our original data, just send us an email along with the relevant source codes. We should be able to share that raw data with you. When we receive a source, we immediately make a record of all the elements that source contains information on, as well as which quads and counties those elements were found in. We distribute this course data through the CNDDB Quick View tool, which is available free to the general public through BIOS. Quick View allows users to query by species and see a map of all the quads where that species is known. Users may also select a county, a quad, or 9 quads to see all the species CNDDB has received data for within those areas. If you query by location, the results show up as a table, as displayed here. This list includes species name, federal and state listing status, and other status information. Specific dates, numbers, and location information are not provided through the Quick View tool. However, it is a simple way to produce a potential species list for pre-project surveys. You can print this table as a PDF or extract it into a spreadsheet. The Quick View tool provides a way to see CNDDB unprocessed information, meaning information not yet mapped into element occurrences. Plants with California Rare Plant Rank 3 or 4 are not actively being mapped as EOs. The Quick View tool would be the best place to find information for those taxa. On the right-hand side of the table, you'll see a data status field. This field tells you if the information is mapped, unprocessed, or mapped and unprocessed. For example, the first row for the snowy plover shows the data status as mapped. This means all the data that CNDDB has received for snowy plover in the Sears Lake Quad has been mapped into EOs. The next 3 rows are for quads with prairie falcon data with status of mapped and unprocessed. That means some of the data is mapped as EOs, but there are additional records that haven't been reviewed. Finally, the second to last row shows the data status for Borrego milkvetch in the Trona East Quad is unprocessed. There are no element occurrences for this species in this quad. Borrego milkvetch is a plant with rank 4, so the CNDDB doesn't map this species as EOs. The Quick View tool would be the best place to see locational information for this species. How do we prioritize our data entry? We record the species, quads, and counties for sources as they are received. However, since we get about 1,000 new sources each month and have a limited number of staff, we need to triage what we map as element occurrences. We generally prioritize our mapping based on anticipated listing activities or other CDFW priorities. That said, if you need an area mapped sooner rather than later, please let us know. You can make a request by emailing the CNDDB coordinator, and we'll do our best to incorporate your request into our workflow. As time permits, we map element occurrences. As mentioned previously, EOs are summaries of everything we know about a species in an area. These include historical accounts of where an element has been known on the landscape, population sizes found, threats in the area, land ownership, ecology, and more. EOs can be used to examine the population viability and imperilment of an element. We don't predict where species may occur when we map element occurrences. Instead, we map what's been reported. Negative data can only be incorporated at previously known occurrences. This means if someone did a survey for an element but couldn't find it, we would only use that negative data if they were looking at a previously known site. So how do we map occurrences at CNDDB? We gather all the data we have for that species and buffer the information with a locational uncertainty. Our methodology dictates that when detections are more than a quarter mile apart, those should represent separate occurrences. We use different types of map features, called graphic accuracy classes, to represent occurrences. Since detections are buffered by some uncertainty, all occurrences are represented by map polygons in the CNDDB. We use standard circle sizes, which range from 80 meters up to 5 miles in radius. It's important to note that the bigger the circle, the greater the spatial uncertainty. Larger circles do not represent more detections of a species. They mean we're less certain of where a detection was made. Our most specific circle has an 80-meter radius. If you provide us with a GPS point, our protocol is to buffer that point by 80 meters. We also have specific and non-specific bounded areas. Specific bounded areas are typically used when the shape of a map feature corresponds to the specific area where the element was found. For example, say someone did a survey for a species of shrub and sent us a GPS point for every individual they found. We could map those precise observations as a specific bounded area around that set of GPS points. Non-specific bounded areas typically indicate a species was found somewhere in the area, but we're not sure where. Say we received information that a bird we track was found nesting in a particular county park. If no specific information was provided, we could map that detection as a non-specific bounded area around the extent of that park. Often, occurrence records will include notes about why an EO was mapped in a certain way. It's always a good idea to read all the text information for an occurrence to get a full picture of what that EO represents. Apart from different accuracy classes, we also use various symbology colors. Green represents plant occurrences, red represents animals, blue and purple are used for aquatic and terrestrial communities. Finally, gray is used for multiples. If you see a gray occurrence, that means multiple elements are mapped on top of one another at that same location. Every occurrence has an occurrence rank. The rank indicates the site condition and species viability for that EO. Occurrence ranks feed into the calculator, which evaluates ranks for entire elements as covered earlier. Occurrences are given letter grades of A for excellent through D for poor, with X representing extirpated. If no information is provided, we use U for unknown. Again, CNDDB staff do not go out in the field to evaluate occurrences. We rely on data submitters to rank the sites that they survey and provide that information to us. Let's show you an example of how we would map an element occurrence and how that occurrence can change over time as we receive more information. EOs are mapped according to the best location information available, but many of the sources used by CNDDB are historic and vague. In this example, a species was documented 50 years ago by a single plant collection. The collection site was described as near Eagle Nest Butte. Originally, the EO was mapped this way, as a large circular feature centered on Eagle Nest Butte. A few years after that, CNDDB received a brief report indicating the plant had been relocated in the same vicinity. The more recent locality was described as near Eagle Nest Butte in Township 45 North, Range 4 East, southwest quarter of Section 30. The EO spatial feature was changed to reflect the more recent and more precise location. The older collection information was retained as a source and mentioned in the text of the occurrence. Further intensive surveys were done after that. Those surveys found the plant in multiple locations in the area. CNDDB ended up receiving 3 datasets reflecting 3 different seasons of fieldwork. This new data included tables of GPS coordinates. CNDDB staff then created small buffers around each of those points and replaced the previous quarter section feature with these specific polygon features. The newest data showed the plant was a little more widespread than known earlier. Since some of these sites are separated by more than a quarter mile with no detections in between, a new separate EO was created to the northeast of the first one. One of CNDDB's goals is to map areas of conservation concern. What this means to a species may vary depending on its biology and life history. A bird spotted while flying over a parking lot may be an interesting detection, but that parking lot may not be biologically significant to the bird. Mapping the parking lot may not be helpful for conservation. Therefore, for highly mobile species, we focus on mapping detections that indicate evidence of reproduction. These can be breeding sites, territorial males, juveniles that can't travel far from the nest, and so on. These sightings usually happen around areas that have biological significance to the species, so it's important to document them. Here's an example of how we would process bird data. First, we receive a data point for a perched bird. Second, we get a report of a flyover. In both cases, the bird may just be passing by. With so little information, we would not assume that these detections are biologically significant, and we would not map them as element occurrences. Then, a few years later, someone submitted data for a nest in a tree. We would now map this nest. The perched bird and flyover that were seen a few years earlier happened in the area, so we would attribute those detections to the occurrence. The EO will be mapped at the nest site, while the EO text will also mention earlier detections nearby. This is another reminder that you should always read the text of element occurrences. Some EO records include extensive notes which aren't obvious to a user who only glances at our map features. Also, feel free to submit your non-nesting bird data to CNDDB. Let us filter the data. We'd prefer to have as much information as possible to work with. From the two previous examples, it should be clear that element occurrences contain information which isn't obvious during a first glance at a map. Users need to read the text to see details on exactly what was detected, when it was detected, how many individuals were found, etc. Our occurrences require interpretation on your part. CNDDB staff take pride in our work. While we strive to be as objective and thorough as possible, we do work under constraints. Let's consider some cases where data collection methods impact how occurrences are mapped. Where a species is found may not be the place it normally lives. For example, these red circles represent a set of badger occurrences. These detections are all along a freeway, but that isn't because roads are prime badger habitat. Badgers are typically nocturnal and difficult to locate. Unless you're actively studying and looking for them, you're likely to find badgers as roadkill, which is how all these detections were made. When looking at such occurrences, be sure to read the text to see what was detected and why. When you take badger behavior into account, you can infer these animals likely had burrows in sandy soil somewhere near the freeway, and they were run over while trying to move from one patch of appropriate habitat to another. Here's another example of sampling bias. This time the bias comes from how people study the species. When biologists perform surveys for California tiger salamanders, they typically visit ponds during the wet season to collect larvae. However, tiger salamanders don't only use ponds. As they become adults, they disperse, moving up to 1 kilometer, they enter burrows, and live for extended periods underground. It's seldom practical to dig through the soil in an area searching for salamanders, so biologists don't normally perform surveys for the underground adults. To preserve tiger salamanders, one can't only preserve their breeding ponds. The surrounding landscape must also be protected. Again, CNDDB maps where detections were made. Our role is not to map the surrounding areas where a species may be found at some future time. If you look at the CNDDB EOs for tiger salamanders, they tend to be centered around breeding ponds because that's the type of survey data we receive. It's up to our users to consider the occurrences in their proper context. Remember, the CNDDB isn't a complete compilation of all locations of rare species in the state. There aren't enough biologists to search all of California. Furthermore, some collected data will never find its way to the CNDDB. Because of this, an absence of data in the CNDDB does not indicate there are no sensitive species in an area. Here's an example. This shows CNDDB occurrences in the Ivanpah Valley in 2006. Here are the occurrences mapped at that same site as of 2020. What changed? Here we have a photo showing how the landscape looked in 2006. And as of 2020, much of this area had been modified by solar energy development. Before construction could begin, detailed biological surveys were required within those areas to be developed. CNDDB received that data and mapped what we received, so these occurrences reflect the level of survey effort. At first glance, someone might get the impression that rare species are more common inside the developed areas than they are in the surrounding landscape, but this is likely a result of where the surveys were performed. Here are some takeaways from the previous example in the Ivanpah Valley. Use a quick view tool to get a list of rare species known to be nearby. Pre-project surveys should be conducted. Don't assume CNDDB is comprehensive. A lack of occurrences may simply reflect a lack of survey effort. If an area has no mapped occurrences in the CNDDB, that fact is not adequate to justify a negative declaration in an environmental document. CNDDB is best viewed as one place to check prior to doing pre-project surveys. In summary, depending on the species, an occurrence may represent many different things. Is it a nest site or a wintering site? Is it a population that's been documented for over 100 years? Was it only known from a single specimen taken decades ago? Were protocol surveys conducted? To know, please read the text. Think about what was mapped and what it means for that species. In many cases, a species may be detected near our occurrences, but not necessarily within one of our mapped polygons. The CNDDB provides a variety of tools and products to help our clients access our data and analyze it for their needs. For free, anyone can access the Quick View tool. For a yearly subscription, you can get full access to our detailed data through BIOS, RareFind, and GIS downloads. As mentioned earlier, the free Quick View tool in BIOS provides course data. You can see a map for a single species across the state. You can also search by location for all track species in a county, a quad, or 9 quads. Those results will include mapped occurrences as well as unprocessed data. QuickView is also the best place to view data on plants with a California Rare Plant rank of 3 or 4. With a paid subscription, you can view CNDDB occurrences using BIOS. You can think of BIOS as a library and CNDDB as a couple of thick books in BIOS. BIOS is a map viewer where you can view thousands of data sets. While most data layers are available to the general public, CNDDB subscribers gain access to additional sensitive species datasets. Subscribers can see our mapped occurrences, the unprocessed data collected through our online field survey form, and other layers with sensitive species data. To view all the text in CNDDB, it's best to use RareFind. RareFind is a website allowing queries and reporting on CNDDB occurrence data. After building a query, you can view the results online or export them in several ways. RareFind and BIOS interlink with one another. You can make a query in one application and pass that query to the other to switch between text and map views. RareFind is built to report data on specific mapped EOs. It allows you to drill down and look at details of occurrences. It does not have access to our coarse unprocessed data. If you need to gather broad data, such as creating a list of all special status species known from a general area, you need to use QuickView rather than RareFind. If you find BIOS doesn't provide the type of spatial analysis your work requires and would prefer to use CNDDB through desktop GIS, we provide GIS downloads through our website. It's important to remember to download the spatial data every month to ensure you're using the latest version. CNDDB data get updated on the first Tuesday of each month. There were 2 versions of the CNDDB data. The government version contains all the data in its original detail, while the commercial version suppresses the exact locations of some EOs. Occurrences may be suppressed because of concerns such as overcollection. Only about 1% of CNDDB occurrences are suppressed. Suppressed data is indicated as quad-sized graphic features. Commercial subscribers can only see that the species occurs somewhere within a particular quad. More detailed information may be given to those with a need to know. Commercial subscribers who are working on a project near a suppressed occurrence may send CNDDB a request for further information along with a map of their project site. Most commonly, the CNDDB is used in environmental regulatory work. Consultants and regulators rely on the CNDDB to inform them about which imperiled species are likely to be found in an area, to glean details about prior surveys, and then determine which types of surveys should be done for a project. Remember that our database is ever-changing and couldn't possibly include every single organism. It is incorrect to assume that a certain project won't impact rare species simply because no occurrences appear on a CNDDB map. Researchers who work with rare species often look to the CNDDB as the best repository of information about where to find their study organisms. CNDDB is used as a major source of information when species are under review for any official listing. Our data also feeds into other CDFW databases and habitat models, including California Wildlife Habitats Relationships, also known as CWHR, and Areas of Conservation Emphasis, known as ACE. CNDDB data contributes to many databases managed by outside organizations, including NatureServe, GBIF, NRIS, CalFlora, CNPS, and others. Natural heritage programs were established to provide decision makers with a one-stop shop for information on rare species data across the Western Hemisphere. Here, over a few seconds, we can see the progression of 40 years of mapping work, which made the CNDDB the largest repository of information on rare species in California. Thousands of people now use our data to inform their decision making. That said, much is still left to discover and share. We encourage anyone with information on rare species to submit their data to us and help fill in the blanks. Now that you know all about how CNDDB works and how you can access and use the information, how do you contribute data to CNDDB? CNDDB is a documentation of the hard work biologists and researchers do in the field. However, we cannot build on this database if we don't ever receive the information. Help us all put the pieces together. If you or your colleagues have rare species data, you may send it to us in a number of formats as detailed on our webpage. Remember, the fastest way to share your data with CNDDB users is through our online field survey form. Our website will walk you through the submission process. For more information, please visit our website, and if you have questions, please feel free to email us.